Dear children, now we are going to see about dark reaction. So far in the previous class, we had discussed about climate reaction in the form of cyclic and non-cyclic photophosphorylation. Now we are going to see about dark reaction. Dark reaction is also called a C3 cycle. Why it is called a C3 cycle? As you know very well that this takes place without involvement of light, hence it is called as dark reaction. This dark reaction, which have been studied by Melvin, Calvin and Benson, that is why this is called as Calvin cycle or Calvin-Benson cycle. As it is producing the first stable product is a 3-carbon compound of phosphoglycerate. This is called as C3 cycle as well. So the dark reaction is also called as C3 cycle or biosynthetic phase or photosynthetic carbon reduction or PCR cycle, or it is also called as Melvin-Calvin cycle. So this biosynthetic phase of photosynthesis is utilizing the assimilated power which are produced during light reaction, that is ATP and NADPH2, which are released during the light reaction. With the help of it, it is reduced the carbon dioxide into carbohydrate. This process is called as dark reaction. As this reaction does not require light energy, this is called as dark reaction. So in this process, RUBP, ribulose 1,5-bisphosphate, is a carbon acceptor to fix the carbon dioxide to form the stable product of 3-carbon compound of phosphoglyceric acid. That is why this is called as C3 cycle. As this was discovered by Melvin Calvin in the year 1957, he was awarded Nobel Prize in the year 1961. Now we shall explain the details of how the Calvin cycle takes place or dark reaction takes place. Usually the dark reaction consists of 3 phases, namely carbon fixation or carboxylation, then reduction process, then finally regeneration of RUBP or ribulose 1,5-bisphosphate. How this takes place, one by one we can see. So in this process, the carbon dioxide acceptor molecule is ribulose 1,5-bisphosphate, RUBP. These are 5-carbon compound. These 5-carbon compound are fixing the carbon dioxide with the help of an enzyme called ribisco to form unstable 6-carbon compound. Then this unstable 6-carbon split into 2 molecules of 3-carbon compound of phosphoglyceric acid or phosphoglycerate. So this is a first stable product which is 3-carbon compound. That is why this is also called as C3 cycle. The cycle is also called as C3 cycle because this is the phosphoglycerate or phosphoglyceric acid is a 3-carbon compound and this is the first stable product. That is why this is called as C3 cycle. Then this phosphoglyceric acid is utilizing ATP which are produced during a light reaction to form 1,3-bisphosphoglyceric acid with the help of an enzyme called kinase. Then the 1,3-bisphosphoglyceric acid is absorbing NADPH2 which are reducing power which are produced during light reaction is also utilized with the help of enzyme dehydrogenase to produce glyceraldehyde 3-phosphate, a stable product of 3-carbon compound of glyceraldehyde 3-phosphate. These glyceraldehyde 3-phosphates are interconvertible with dihydroxy acetone phosphate. These are also 3-carbon compound. These are mutually transferable by the enzyme called triose phosphate isomerase. With the help of triose phosphate isomerase, glyceraldehyde 3-phosphate and dihydroxyacetone phosphates are mutually interconvertible. Some of the glyceraldehyde 3-phosphate is converted into dihydroxyacetone phosphate will be export out. Then the glyceraldehyde 3-phosphates are combined with dihydroxyacetone phosphate with the help of an enzyme called aldolase to form fructose 1,6-bisphosphate. So glyceraldehyde 3-phosphate and dihydroxyacetone phosphate both are 3-carbon compounds. So two 3 carbon compounds combine together to form a 6 carbon compound called fructose 1,6,6 bisphosphate. Bisphosphate means 2 phosphate. One phosphate is attached at the first position, second phosphate is attached at the 6th position of 6 carbon. So this is called as fructose 1,6 bisphosphate. Out of this fructose 1,6 bisphosphate, the first position of the phosphate is dephosphorylated with the help of enzyme called phosphatase. Then it is forming fructose 6 phosphate. So fructose 6 phosphate entered into the stroma hexose phosphate pool. These are mutually interconvertible isomerase. That means fructose and glucose are isomerase, so they can be able to form fructose 6 phosphate into glucose 6 phosphate. Similarly, glucose 6 phosphate is converted into glucose 1 phosphate as well. Finally, they can be able to convert it into starch. Some of the fructose 1,6 bisphosphate, fructose 6 phosphate is combined with glyceraldehyde 3 phosphate. So this is 6 carbon compound, this is 3 carbon compound, totally 6 plus 3, 9 carbon compound, which will be split into 2 molecule called xylose 5 phosphate. This is 5 carbon compound, erythrose 4 phosphate, which is 4 carbon compound with the help of enzyme called transketolase is formed. Then this 4 carbon compound of erythrose 4 phosphate is combined with 3 carbon compound of dihydroxyacetone phosphate. 3 plus 4, 7. That is pseudoheptose 1,7 bisphosphate with the help of enzyme aldolase is formed. This pseudoheptose 1 phosphate is a 7 carbon compound which are having phosphate at first position and 7th position. Then the first position phosphate is dephosphorylated to form pseudoheptose 7 phosphate. This is 7 carbon compound. This pseudoheptose 7 carbon compound or pseudoheptose 7 phosphate is combined with 3 carbon compound of glyceraldehyde 3 phosphate to form 10 carbon compound to split into 2 5 carbon compound of xylose 5 phosphate and ribose 5 phosphate with the help of enzyme called transketolase. Then here the ribose 5 phosphate is isomerized with to form ribulose 5 phosphate. Then xylose 5 phosphate, which is formed from fructose 6 phosphate, which will be converted into ribulose 5 phosphate with the help of enzyme epimerase. Here it is formed xylose 5 phosphate here also. That is also converted into ribulose 5 phosphate by the enzyme called epimerase. Then ribulose 5 phosphate is phosphorylated with the ATP with the help of kinase enzyme to form once again regeneration of RUBP. That is ribulose 1,5 bisphosphate. So here carbon fixation, here all reduction, here the regeneration. The 3 processes called as regeneration of RUBP is occur. So this is called as Calvin cycle. This Calvin cycle takes place without light energy. That is why these are called as dark reaction. Now we are going to see another concept. So far we have discussed about both light reaction as well as dark reaction in detail. Now we are going to discuss about C4 pathway or Hatch-Lock pathway. C4 cycle is also called as Hatch-Lock pathway. This is also called as dicarboxylic acid pathway or dicarboxylation pathway. What is Hatch-Lock pathway? There are 2 important scientists who are named as Hatch and Slack. In the year 1967, both have been observed that in certain plants like sugarcane, maize and some other monocot plant, they are having another type of carbon fixation. So far we have seen about Calvin cycle. It is C3 pathway. Why it is C3 pathway? The first stable product is 3 carbon compound of phosphoglyceric acid. That is why these are called as C3 pathway. So Hatch and Slack, the 2 scientists who have studied about some of the plant, they have observed that one more pathway. After fixation of carbon, the first stable product is 4 carbon compound That is why these are called as C4 cycle. This observation was confirmed by Hatch and Slack in the year 1967. This is an alternate pathway for the fixation of carbon dioxide was found in several tropical and subtropical grass plant and some dicots. Usually, C4 cycle is discovered in more than 1,000 species. Among them, 300 species belongs to dicots and the remaining rest of the plants are monocots. C4 plants represented about 5% of earth plant biomass and 1% of its known plant species. So C4 pathway is completed in two phases. That is, first phase which takes place in mesophyll cells. The second phase which takes place in bundle sheath. Look at the picture. If we take a cross section of leaf, so this is outer epidermis, then green color mesophyll, then vascular bundle. The vascular bundle is surrounded by bundle sheath. So here the C4 cycle takes place in two carbon fixation. One carbon fixation takes place in mesophyll cell. So this is mesophyll cell. The second carbon fixation takes place in bundle sheath. So this is second carbon fixation. So this is called as C4 cycle or Hatch-Watt pathway. Here the carbon fixation which takes place by phosphoenol pyruvate or phosphoenol pyruvic acid. This is 3 carbon compound. This 3 carbon compound of phosphoenol pyruvic acid fixing atmospheric carbon dioxide to form 4 carbon compound of oxaloacetic acid with the help of an enzyme called phosphoenol pyruvic acid carboxylase, PEP carboxylase. So PEP is absorbing carbon dioxide from the atmosphere in mesophyll cell with the help of PEP carboxylase to produce oxaloacetic acid. This is the first stable product. It is 4 carbon compound. That is why this is called a C4 cycle. Then this oxaloacetic acid will be converted into either malic acid or aspartic acid. Both malate and aspartate are 4 carbon compound. Then this malate will be entered into the bundle sheath. Then they can able to split into pyruvic acid and carbon dioxide. The pyruvate, once again it is returned back to the mesophyll cell to form phosphoenol pyruvic acid. But this carbon dioxide is entered into the Calvin cycle to form sugar. This sugar is entered into the vascular tissue. So this cycle is called as C4 cycle or Hatch-Watt pathway. So this is a great picture which is given by Hatch and Slack. Now one important thing about one of the important question is what are the differences between C3 cycle and C4 cycle? That is differences between Calvin cycle and Hatch-Watt pathway. In C3 cycle, the carbon fixation takes place only in mesophyll cells. But in C4 cycle, the carbon fixation takes place in mesophyll as well as bundle sheath. So the carbon fixation takes place mesophyll and bundle sheath in C4 cycle. In C3 cycle, carbon acceptor is ribulose bisphosphate, RUBP. So in C3 cycle, RUBP is the only carbon dioxide acceptor. But in C4 cycle, phosphoenol pyruvic acid, PEP, is a carbon acceptor in the mesophyll cell, and Calvin cycle, that is RUBP, is a carbon fixation in the bundle sheath. So there are 2 carbon fixation. One is by phosphoenol pyruvic acid, another one is by RUBP in Calvin cycle. Then the first product in C3 cycle is phosphoglyceric acid. This is 3 carbon compound. But here the first stable product is 4 carbon compound of oxaloacetate. Trans anatomy is not present, but here Grant's anatomy is present. So, which is showing the Grant's anatomy. Actually, in C3 cycle, only one type of chloroplast is there. But in C4 cycle, there are two types of chloroplast. That is called dimorphic chloroplast. That means the chloroplast present in the mesophils are having with granule. That is called granule chloroplast. But the chloroplast present in the bundle sheath, they do not have any granule. That means a granule chloroplast. So there are two types of chloroplast are present in C4 cycle. So one is granule chloroplast in mesophil cell and a granule chloroplast in bundle sheath. So this is called as dimorphic chloroplast. So dimorphic chloroplast are present in C4 cycle, but in C3 cycle, granum is present in mesophil. And moreover, they are having normal chloroplast, but in C4 cycle, it is having dimorphic chloroplast. Then optimum temperature is required for the C3 cycle is around 20 degrees Celsius to 25 degrees Celsius. But for C4 cycle, the optimum temperature is 30 degrees Celsius to 45 degrees Celsius. The fixation of carbon dioxide is at 550 parts per million in C3 cycle, but fixation of carbon dioxide even less than 10 parts per million takes place in C4 cycle. C3 cycle is less efficient due to higher photophosphorylation takes place. But in C4 cycle, photophosphorylation is very minimum. So that is why this is more efficient due to less photophosphorylation. RuBP carboxylase enzyme is used for carbon dioxide fixation in C3 cycle, but PEP carboxylase enzyme and RuBP carboxylase enzyme are replaced for carbon fixation. There are about 18 ATPs are used to synthesize one glucose molecule in C3 cycle, but in C4 cycle, they can consume 30 ATPs to produce one glucose. C3 cycle is efficient at low carbon dioxide. C4 cycle is efficient at high carbon dioxide. Lot of example can be seen for C3 cycle like paddy plant, wheat, potato and so on will be very good example for C3 plant. But C4 pathway takes place in sugarcane, maize, sorghum, amaranthus. Amaranthus means kiri, totakiri, tribulus. These are all example for C4 plants. That's all about the differences between C3 and C4 cycle. Now we are going to see about another mechanism is called as Kame cycle or granulation acid metabolism cycle. That is Kame metabolism. Look at this picture. This picture will be helping us to understand about Kame metabolism. Kame cycle. Kame cycle means granulation acid metabolism. So usually we have seen about either carbon fixation, which will be takes place either C3 or C4 cycle. So due to carbon fixation, the stomata is closed and open, processes takes place. But certain conditions of zero fatty condition in desert or dry or drought area. So lot of succulent plants might be there. Especially a family belongs to Gracinaceae family. A special type of cycle takes place. That is why these are called as Gracilasian acid metabolism cycle. That is Kame cycle. So it is one of the carbon pathway identified in succulent plant. Succulent means the leaves are very fleshy. Growing in semi-arid. Semi-arid means dry or zero fatty condition. This was first observed in Gracilasiae family plant like Breophyllum, Cattipatal Puttipodum, as you know, a thick fleshy leaf. Then Sedum, Kalancho. And this is the reason behind the name of the cycle. It is also noticed that in plant from other families such as aloe vera, agave, opuntia, pineapple, and certain orchids are also doing this Kame metabolism. So usually all the plants are opening the stomata during daytime and closed at night. But these plants are opening the stomata during night and closing the stomata during daytime. How this mechanism takes place? Such a type of mechanism is called cam cycle. So this is a reverse stomatal rhythm, helps in conserve the water loss through transpiration and will stop the fixation of carbon dioxide during daytime. That means if they are opening during daytime, the excess amount of water will be evaporated due to the process of transpiration. Such a type of things can be prevented by this method. So during night time, the stomata is open. Then what happens? The atmospheric carbon dioxide is entered inside. Then excess amount of water is also evaporated. But during the transpiration rate is minimum. So uptake of carbon dioxide takes place. This uptake of carbon dioxide takes place by phosphoenolpyruvic acid, PEP, with the help of enzyme PEP carboxylase. Originally the PEP is formed from starch. Then the starch will be converted into triose phosphate in chloroplast of cam plant, then which is producing phosphoenolpyruvate. Then this phosphoenolpyruvate is accepting the atmospheric carbon dioxide with the help of PEP carboxylase enzyme to form 4-carbon compound of oxaloacetic acid like C4 pathway. Then this oxaloacetic acid is converted into malic by NAD plus malic dehydrogenase enzyme. Then this is producing malic acid in vacuum. So which within the vacuum, this malic acid is stored. So this takes place in stomata. So hence the carbon dioxide is accepted, then it is dissolving to form acidic condition. Then the stomata is open. Then it is absorbing water and they become turgid, the stomata is open. But during daytime, the stomata is closed. Prevent loss of water as well as entry of carbon dioxide is also prevented. So this decarboxylation takes place. The carbon dioxide is not entered because of the stomata is closed. But even though carboxylation takes place, how? Already the malic acid is having converted into malic acid and break down to produce carbon dioxide and pyruvic acid. So this decarboxylation takes place from the malate by the enzyme called malate enzyme. Then this will be fixed by Calvin cycle. This type of process takes place during night. So during night time, they can do the process of photosynthesis. That means fixation of carbon dioxide takes place during night. So this is a special type of mechanism which takes place in certain plants like Gracilaceae family. So this will stop the fixation of carbon dioxide during daytime. At night, cam plants can fix carbon dioxide with the help of phosphoenolpyruvic acid and produce oxaloacetic acid, 4-carbon compound. Subsequently, oxaloacetic acid is converted into malic acid like C4 cycle and get accumulated in vacuum, increasing the acidity. So during the daytime, the stomata are closed. And malic acid is decarboxylated into pyruvic acid, resulting in degrees of acidity during daytime. So carbon dioxide thus formed enter into the Calvin cycle and produce carbohydrate. So this is called cam metabolism or cam cycle. Now we are going to see another important concept which is called as photorespiration. The photorespiration is also called a C2 cycle or photosynthetic carbon oxidation cycle. This can be seen with the help of this picture. So this is C2 cycle or photophosphorylation cycle. Already you know that what is respiration. The details of respiration process we can see in the next chapter, tomorrow class and next class. What is respiration? Breaking down of glucose with the help of oxygen to produce carbon dioxide, water and energy. This process takes place in the presence of oxygen is called aerobic respiration. If the process takes place in the absence of oxygen to form either lactic acid or ethyl alcohol, which will be called as anaerobic respiration. So both aerobic as well as anaerobic respiration might be takes place in living organisms. This is normal respiration. But so this is another type of respiration which is called as photorespiration. So usually respiration do not need any sunlight, but this respiration takes place only in the presence of photosynthesis. Only in the presence of light, and moreover, this photorespiration process which takes place in photosynthetic organelles like chloroplast, peroxisome, mitochondria. So respiration is a continuous process for all living organisms, including plants. A great scientist, Decker, in the year 1959 observed that the rate of respiration is more in light than in darkness. That is photorespiration. So photorespiration is the excess respiration taking place in photosynthetic cells due to the absence of carbon dioxide. So if there is no carbon dioxide, then the photorespiration only takes place. And increase the oxygen. This condition changes the carboxylase role of ribosco into oxygenase. So this is C2 cycle. Why it is called a C2 cycle? The first stable product is 2-carbon compound of phosphoglycolate or phosphoglycolic acid. This is 2-carbon compound. This is first stable product. That is why this photorespiration process is also called as C2 cycle. So C2 cycle or photorespiration takes place in chloroplast, peroxisome and mitochondria. Here, how this process takes place, we shall see one by one. In chloroplast, RUBP, ribulose 1,5 bisphosphate, is oxidized with excess amount of oxygen. Ribulose 5-phosphate is 5-carbon compound, which is oxidized with excess amount of oxygen to produce phosphoglyceric acid and phosphoglycolate with the help of enzyme called ribosco. So the PGA, phosphoglyceric acid, enter into the Calvin cycle. The remaining molecule is phosphoglycolate. Then this phosphate molecule is dephosphorylated to produce glycolate in chloroplast. Then this glycolate is entered into another organelle called peroxisome. Then this glycolate is oxidized. So which is utilizing more amount of oxygen to form glycolate into oxalate as well as hydrogen peroxide. This hydrogen peroxide may produce water and oxygen. Then this glyoxalate is producing glycine in peroxisome. Then this glycine is entered into the mitochondria. When two molecules of glycine combine together to form another amino acid is called serine. Here in this process, carbon dioxide is evolved and ammonia is evolved. Then serine is entered into the peroxisome from mitochondria. Then serine is converted into hydroxypyruvic acid. Then hydroxypyruvic acid is oxidized from NADH2 is converted into NAD plus to form glyceric acid or glycerate. Then this glyceric acid is entered into the chloroplast again. Then the glyceric acid is phosphorylated with ATP to form phosphoglyceric acid. The phosphoglyceric acid entered into the Calvin cycle. Once again, the ribulose 1,5 bisphosphate is formed. So this process is called as photorespiration process. This photorespiration process takes place in some plant. Then what is the significance of this photorespiration? Some of the amino acids like glycine, serine, synthesized during this process are precursor of many biomolecules like chlorophyll, proteins and nucleotides. It consumes NADH plus H plus and generated. Glycolic acid protects cell from photooxidation. So excess amount of sunlight might not be affect the plant by the process of photooxidation. So from the photooxidation, this photorespiration process will be protecting the plant. So another important question is, what are the differences between photorespiration and dark respiration? Usually the photorespiration takes place in photosynthetic green cells, but dark respiration takes place almost all living cells. Photorespiration takes place only in the presence of light, but the dark respiration takes place at all time. They do not require any light. Photorespiration involves chloroplast, peroxisome and mitochondria, but dark respiration is involved only in mitochondria. Photorespiration does not involve in glycolysis, Krebs cycle and electron transport system that can be studied in the next chapter in respiration. But dark respiration is involving with glycolysis, Krebs cycle and electron transport system. Substrate of photorespiration is glycolic acid, but substrate of dark respiration is carbohydrate, proteins and fats. Photorespiration is not essential for survival, but respiration is very much essential for each and every living organism, because all the organisms obtain energy from respiration. There is no phosphorylation or ATP is synthesized during photorespiration, but ATP is synthesized during dark respiration by oxidative phosphorylation process. In photorespiration, NADH2 is oxidized. So here you might have seen that. Here NADH2 is oxidized to NAD+. But in dark respiration, NAD is reduced to NADH2. In photorespiration, hydrogen peroxide is produced here. This is hydrogen peroxide, H2O2 is hydrogen peroxide. So hydrogen peroxide is produced during photorespiration, but hydrogen peroxide is not produced during dark respiration. End product of photorespiration is carbon dioxide and PGA, but end product of dark respiration is carbon dioxide and water. So these are the main differences between photorespiration and dark respiration. Now we are going to see about factors affecting photosynthesis. Different types of factors which will be responsible for the process of photosynthesis. If these factors are available, then the process of photosynthesis might be takes place. Such a type of factors will be called as factors affecting photosynthesis. One by one we can see lot of factors which are involved. Basically, among the factors, some of the factors are very much essential. Factors affecting photosynthesis are further classified into external factors and internal factors. Some of the factors are available inside the plant itself. They are called as internal factors like pigments, protoplasmic factor, accumulation of carbohydrate, anatomy of leaf and hormones. External factors are light, carbon dioxide, then temperature, water, minerals and various types of pollutants are also involved. Among them, one of the important factors is Blackman's law of limiting factors. What is Blackman's law of limiting factor? When a process is conditioned as reduced rapidly by number of separate factors, the rate of process is limited by the pace of lower factor. That means at any given point of time, the lowest factor among the essential will limit the rate of photosynthesis. For example, both carbon dioxide and sunlight are essential. Only in the presence of carbon dioxide and water and chloroplast, the photosynthesis might be takes place. Sometimes there is a failure of sunlight. Suppose if the weather is cloudy, the sunlight is minimum. Then what will happen? The factor which is very less will be determining the rate of photosynthesis. That means here sunlight is very limited. That is why the sunlight is determining the rate of photosynthesis. Suppose in the atmosphere carbon dioxide is very less. Then what happens? The carbon dioxide is a determining factor. So this is called law of limiting factor. So law of limiting factor is nothing but at any given time, the lowest factor among the essential will be limit the rate of photosynthesis. For example, when even sufficient light intensity is available, photosynthesis may be low due to low carbon dioxide in the atmosphere. Here, carbon dioxide acting as a limiting factor. Suppose if carbon dioxide is increased in the atmosphere, the rate of photosynthesis also increases. Further, increase in photosynthesis is possible only availability of light intensity. Now one by one we can see. First of all, we can see about various internal factors like light, temperature and so on. So light is very important factor. Whenever the intensity of light is available, the rate of photosynthesis gradually increases. Especially when the synthetically active radiation power around 400 to 700 nanometer, the photosynthetic rate will be more. So such a type of wavelength will be called as the photosynthetic active radiation. So availability of light will be depends upon the rate of photosynthesis. If they increase the intensity of light, the rate of photosynthesis also maybe increases, but up to certain level. But if it is increasing more than its limits, which may affect the chlorophyll molecule, then the rate of photosynthesis may be reduced. Then availability of carbon dioxide. So carbon dioxide found only around 0.3% in the atmosphere. But even though it is very limited, they play a vital role in the rate of photosynthesis. So increase in the concentration of carbon dioxide, that will proportionally increase the rate of photosynthesis up to 330 parts per million. But beyond 500 parts per million of carbon dioxide, the rate of photosynthesis will be unaffected. That means remain constant. Then availability of oxygen. The rate of photosynthesis will be decreases when there is an increase in oxygen concentration. This inhibitory effect of oxygen was first discovered by Wasberg using green algae called chlorella. Then temperature. The optimum temperature for the photosynthesis vary from plant to plant. So, but optimum temperature is required for the process of photosynthesis. So, temperature is not uniform in all places. In general, the optimum temperature for the photosynthesis is around 25 degrees Celsius to 35 degrees Celsius. But this is not applicable for all plants. Ideal temperature for plant like Opensia is 55 degrees. Suppose lichen is having 20 degrees Celsius and algae growing in hot spring photosynthesis is around 75 degrees Celsius. So, depends upon the requirement of plant, the variations of temperature is required for the plant. Then water. Water is a very important role in process of photosynthesis. Photolysis of water takes place during non-cyclic photophosphorylation pathway. So photolysis of water provide electrons, protons for the reduction of NADP as well as directly or indirectly role in stomatal movement. So during water stress, the supply of NADH plus H plus is also going to be affected. Then various minerals also doing an important role affecting the photosynthesis. Deficiency of certain minerals affect the photosynthesis. For example, minerals involved in the synthesis of chlorophyll is magnesium, iron and nitrogen. So phosphorylation reaction that is takes place with the help of phosphorus. Photolysis of water will be depends upon manganese and chloride ion. Then formation of photocyanide is copper. So in such a way depends upon various processes, different types of minerals required. Some of the air pollutant is also affecting photosynthesis. Some of the pollutant like sulfur dioxide, nitrogen dioxide, ozone and smoke affect the rate of photosynthesis. Now we are going to discuss about internal factors. Photosynthetic pigment, it is essential factor and even a small quantity is enough to carry out photosynthesis. So all the pigments like chlorophyll A, chlorophyll B, chlorophyll C and so on, all the pigments are essential, but even a minimum quantity is required to complete the process. Then protoplasmic factor. The hydrated protoplasm is essential for photosynthesis. It also includes enzymes responsible for the photosynthesis. Then accumulation of carbohydrate inside the cell. So photosynthetic end product like carbohydrates are accumulated inside the cell, and if the translocation of carbohydrate is slow, then this will affect the rate of photosynthesis. Then anatomy of leaf, internal structure of leaf. So thickness of cuticle and epidermis, then distribution of stomata, presence or absence of plant's anatomy, and relative proportions of photosynthetic cells affect the photosynthesis. And some of the important hormones like gibberellin and cytokinin also increase the rate of photosynthesis. Now there is an important experiment, that is, how do you, already we know that during photosynthesis process oxygen is evolved. How do you know that, or how do you prove that oxygen is evolved during photosynthesis? This can be done with the help of one experiment is called test tube and funnel experiment. This experiment will be helping us to prove oxygen is evolved during photosynthesis. This can be seen with the help of this experimental setup. This experimental setup will be helping us to understand about how the oxygen is evolved. Look at this picture. So this is hydrolyte twigs. These are kept in a glass funnel which are kept in inverted position. Then filled with water, a test tube is filled with water and inverted over the stem of the funnel. That should be kept in a beaker containing water. That should be kept in sunlight for a few hours. Then we can identify how the oxygen is evolved during photosynthesis. Place some hydrolyte plant at the bottom of the beaker containing water. Some hydrolyte twigs to be kept inside the thistle funnel, glass funnel, in inverted glass funnel. Cover the plant with an inverted funnel. Then fill a test tube and invert over the stem of the thistle funnel. Keep the setup inside the beaker containing water. Then the setup should be kept in sunlight. After some time we can observe that air bubbles are released from the hydrolyte twigs. Then these air bubbles are collected at the end of the tube. Then carefully remove this test tube and bring a burning splinter. The burning splinter burns brightly. This indicates that the evolution of gas is oxygen. So from this experiment we can conclude that oxygen is evolved during photosynthesis. Now we are going to see about photosynthesis in bacteria. So far we had seen about photosynthesis in common plant. Now we are going to see about photosynthesis in bacteria. Though we are studying about the bacterial photosynthesis at last part of this lesson, actually bacterial photosynthesis formed first and foremost in the evolution. Bacteria does not have any specialized structure like chloroplast, but in higher plants they are having a prominent chloroplast which is doing photosynthesis function. But in bacteria, there is no chloroplast at all. But even the photosynthesis processes takes place. How? It has simple type of photosynthetic apparatus called chlorosomes and chromatophore. These two important organelles which will be helping the bacteria to doing photosynthesis. Van Neel in the year 1930 discovered a bacterium that releases sulfur instead of oxygen. So all the higher plants are releasing oxygen. So this experiment will be helping us to understand that oxygen is evolved during photosynthesis. But during bacterial photosynthesis, there is no evolution of oxygen. Van Neel discovered that bacterium is releasing sulfur instead of oxygen during photosynthesis. And here the electron donor is hydrogen sulfide. But in higher plants we have seen only one photosystem is involved. And moreover, in bacterial chloroplast, in bacterial photosynthesis, only one type of photosystem is involved. But in higher plants, photosystem 1 and photosystem 2. But in bacteria, the photosystem 1 is involved. And moreover, the reaction center is P870 nanometer. And moreover, in higher plants, we used to see chlorophyll A, chlorophyll B and so on. But specialized pigments are present in bacteria are called bacterial chlorophyll. Bacterial chlorophyll A, bacterial chlorophyll B, C, D and even G. Some of the carotenoids are also present. Generally, the photosynthetic bacteria are classified into three different categories like green sulfur bacteria, purple sulfur bacteria, purple non-sulfur bacteria. For example, Chlorobacterium, chlorobium comes under green sulfur bacteria, Thiospirillum and Chromatium which will becomes under purple sulfur bacteria. Then Rhodopseudomonas, Rhodospirillum, these are comes under purple non-sulfur bacteria. Now there is an important question is, what are the differences between photosynthesis in plants and photosynthesis in bacteria? If you see, in plants, the cyclic and non-cyclic photophosphorylation takes place. But in bacteria, only cyclic photophosphorylation takes place. In plants, both photosystem 1 and photosystem 2 is involved. Two are involved. But in bacteria, only photosystem 1 only involved. In plant, electron donor is water. But in bacteria, electron donor is hydrogen sulfide. In plants, oxygen is evolved. But in bacteria, oxygen is not evolved. Reaction center in plants, P700 and P680, that is photosystem 2 and photosystem 1. But in bacteria, reaction center is P870, photosystem 1. Reducing agent like NADP H plus is in plant, but reducing agent in bacteria is NADH plus H plus. Photooxidation radiation, that means photosynthetic active radiation is around 400 to 700 nanometer, but in bacteria 700 nanometer. Chlorophyll, carotenoids, xanthophylls are the pigments which are present in plants. But in bacteria, bacterial chlorophyll and bacteria viridin are the pigments. Photosynthetic apparatus of plants are chloroplast, but in bacteria, chlorosomes and chromatophores are the organelles for the photosynthesis. That's all about the differences between photosynthesis in plants and bacteria. That's all about photosynthesis. Thank you.